Biology for 10th standard. Plant anatomy. Today we are going to learn about the internal structure of dicot and monocot stem. First of all, before we are going to see the internal structure, there are the basic concepts on monocot and dicot. We need to learn the difference between monocot and dicot. So these are the few characters of dicot and monocot. First, monocots. So seeds of monocotyledon will be a single cotyledon and the leaves will be having parallel veins. Then the flowers will be in multiples of 3. That means 3 petals or multiples of 3. Then the vascular tissues are scattered throughout the stem. Then the root system is fibrous root system. It bears very minute hair-like fibrous root systems. Then the next one is dicots. So the dicot plants arise from 2 cotyledons and the leaves are branched veins. Then the flowers are often in multiples of 4 or 5. That means it bears 4 or 5 petals. Then the next one is vascular tissues, which are arranged in a ring-like structure. Then the root system is taproot system. That means the very strong, deep root system will be present in dicots. Before we get into the internal structure of stem, we need to learn the important terms like epidermis, cortex, endodermis, pericycle, and the types of xylems like endarch and exarch, then pith. So these are the important terms we need to learn before. The first layer is called epidermis, outer covering layer, which covers and protects the plant's surface. It secretes a waxy substance called cuticle. When plant matures, epidermis is replaced by a layer called periderm. The next one is cortex. It is a region which lies between epidermis and vascular tissues. Cortex is made up of mostly parenchymatous tissue, which stores carbohydrates, latex, oils, resins, etc. Next, endodermis, which is next to cortex, and it is the outer covering layer of vascular tissues. It consists of Casparian stripes. It is a cell wall material. And the next one is pericycle, which lies just below the endodermis and which is made up of parenchyma and sclerenchymatic cells. Then the next part is vascular tissues. As you already know that xylem and phloem are called vascular tissues. The arrangement of xylem and phloem are different between monocot and dicot plants. There are 2 types of xylem, endarch and exarch. The first one is endarch xylem. Endarch means it is the arrangement of protoxylem towards center and metaxylem towards outer, whereas exarch is just opposite to endarch, where protoxylem towards the outer and metaxylem towards the inner side. First of all, protoxylem means primary xylem or first xylem form. Then when it matures to form a metaxylem. Basically, protoxylem looks small when compared to metaxylem. So based on the presence of protoxylem and metaxylem, it is divided into 2 types as endarch and exarch. So the last part is called pith, which is the center of the stem, made up of spongy parenchyma, which stores and transports the nutrients. So these are the few important terms which are essential for learning the internal structure. So now we are ready to learn the internal structure of dicot stem. So here is the picture of dicot stem. So the first layer is epidermis. So epidermis is meant for protection. It consists of cuticle and epidermal hairs. Then the next layer is hypodermis, which is below the epidermis, and it is made up of collenchymatous cells. The next one is ground tissue or cortex region, which is made up of chlorenchymatous and parenchymatous cells. Chlorenchymatous means nothing but the cells which possess chlorophyll is called as chlorenchymatous cells. Then the next tissues is called endodermis. So it is a layer which is outer to vascular tissues. Then below the endodermis, pericycle appears. The next to pericycle, the vascular tissues called xylem and phloem are arranged in a ring-like structure. This ring-like structure is a special characteristic of dicot stem. And Here the type of vascular bundle is conjoined, collateral, open-end or vascular bundle. You know what is meant by conjoined, collateral. Open means presence of cambium which gives rise to secondary xylem and phloem during secondary growth. And end arc xylem. End arc means protoxylem will be towards the center. So this is a type of vascular bundle which is present in the dicot stem. So these are the characteristics and internal structure of dicot stem. So the next one is monocot stem. Here also the first layer is epidermis, which is meant for protection, and it also consists of a layer called cuticle. The next one is which is below the epidermis is called as hypodermis, which is made up of skeleton chimatous cells. Then the next layer is called cortex. Here in monocot stem, the cortex is not differentiated into endodermis, pericycle and pith when compared to dicot stem, because the vascular tissues are scattered in the monocot stem. Then the type of vascular tissue is as like dicot, here also conjoined, collateral but closed vascular system. That means cambium will be absent. It doesn't give rise to new xylem and phloem. So these are the characteristics of internal structure of monocot stem. So here is the slide for your summary.